In a previous talk at the Tokyo Polytechnic University's symposium, we discussed vision and color vision in the real natural world. In that talk, we didn't spend much time on color constancy, a favorite topic of my research. And so this is a second video that specializes just on color constancy. It talks about how edges, not objects' reflectance, is the important thing in understanding why colors remain the same in different illumination. In the real world, we have an illumination source that falls on an object. A fraction of that light gets reflected and imaged by our lens onto our retina. In the retina, we have receptors that capture the photons and give us the receptors' quanta catch. That is the signal that is sent on to our brains. There's a long history about light-matter interactions and about quanta catch. It began with Newton, Leblond, who did the first color printing, Thomas Young, who invented trichromacy, and Maxwell, who established colorimetry. In 1870, in Germany at least, it was known as the Young-Helmholtz theory. Helmholtz was a leading vision expert in many fields, and he was the major proponent of how quanta catch controlled color matches and controlled color appearances. However, there's a different way of studying the world and color. In that world, we have a complex scene in which the object of interest is just one of many things in the field of view. Again, illumination falls on the object, but it's in a rich, complicated surround. All the radiances are imaged on the retina, and all the receptors give you a quanta catch signal as the input to vision. The second approach to vision has been studied by artists, Da Vinci, Goethe, Chevreul, Herring. These students of color are more interested in the color constancy in the real HDR world. They claim that appearance is not equal to the quanta catch of the receptors. So in 1870, we have a problem. We have a group of distinguished scientists who say that the quanta catch of the receptors in the retina controls color vision. We have a different group of color experts that say the quanta catch can't be the right thing because colors are constant despite the fact we live in a world of changing illumination. We have a dispute between the light-matter interaction color experts and the appearance color experts. Now that debate has continued for about 150 years. In 1872, Ewald Herring wrote, "The approximate constancy of colors of seen objects, in spite of large quantitative and qualitative changes in the general illumination of the visual field, is one of the most notable and most important facts in the field of physiological optics. Without this approximate constancy, a piece of chalk on a cloudy day would manifest the same color as a piece of coal does on a sunny day. In the course of a single day, it would have to assume all the possible colors that lie between black and white. Similarly, a white flower that is seen under a green arbor roof would show the same color as a green leaf under open sky. And a ball of yarn, white in daylight, in gaslight, would have the same color as an orange." Herring was the proponent of color constancy. At the same time, Helmholtz added to the colorimetrist Young Helmholtz theory, the second idea of unconscious inference. He wrote, by seeing objects of the same color under these various illuminations, in spite of difference of illumination, we learn to form a correct idea of the color of bodies. The champion of bottom-up quanta-catch retinal response says that's not important. What's important is the object's reflectance. If we want to obtain the information about the object's reflectance, we need to discount the illumination. And we do this by unconscious inference. Somehow or other, the brain unscrambles the light caught by the receptors in the retina and applies this unconscious inference to inform us about the object's reflectance. To say this debate's been going on for 150 years, a recent paper, Helmholtz had been too ready to relegate sensory processes to the mental sphere and give up the search for physiological explanation. The same author wrote, Herring would be pleased with the growth of our knowledge of receptive fields of retinal and cortical cells, and of the structure of lateral connections that possibly relate to contrast effects and in our ability to detect connected edges. This was written in 1999. So, 1870 color is built up of light-matter interactions that go bottom-up, Newton, Young, Helmholtz, or a top-down approach that recognizes objects, uses Helmholtz unconscious inference, influenced by Herring and Goethe and the color constancy proponents. But note, we have Helmholtz in both traditions. This is a big change for a physicist. It shifts the problem from being a matter of physics to a matter of recognition of higher cortical processes. This is a big change. This is a really big change. How can we sort this out? In 1967, Land performed a very important experiments using two color Mondrians. This experiment was first presented at Land's Ives Medal Address to the Optical Society of America. It consisted of having two 1.5 meter square displays of colored papers, 3 illuminating projectors for the left Mondrian, 3 different projectors to illuminate the right Mondrian, means to control the amount of output of the projectors, and means to measure the amount of light from a certain spot. This is a photograph of Edwin Land, a famous inventor, businessman, and color scientist. This is a photograph of him at another Optical Society meeting where he introduced instant photography. Probably the best paper in explaining his Retinex theory is a paper given at the Royal Institution in 1974. There's a link to that here. And in it, there's a drawing done by Gene Benton. There's Land giving the talk, and that's me adjusting all these projectors and doing the demonstration. My job was to measure the amount of light that came from this green circle. We had a meter and we projected the numbers up on the top of the screen. Then I would go to the second Mondrian and adjust the amount of light coming out of these 3 projectors so that the amount of light at the tip of this arrow was identical to the amount of light at this arrow. Land was very careful. He made us do things like check that the meter was right. And so if the meter was right and these were identical stimuli, if you looked at these without anything else in the field of view, you would have the same stimulus here and the same stimulus here, and without anything else, they have to look identical. So if you looked at these separately, you'd see two spots of light. They'd be the same. They are not red and they are not green. When you exclude everything in the scene except these two circles, you see two identical colors. Curiously, they are neither red nor green. They appear identical. When we go to the real life situation of seeing the entire field of view, they look different. However, if they're in the world of colorimetry, a spot and no light around it, they will look the same. The reason is the cone quanta catches are the same. So we have a match in this situation, but when we go to the real world, they need not be the same. Let's study this in a little bit more detail. Here we have the two Mondrians. Now remember, my job was to make the light from this circle be identical to the light from this circle. I did it by changing the illuminations uniformly, but this illumination was different than this illumination. So if we look at the green circle, green is a poor reflector of long wave light so that it looks dark in relation to the others. Over here we have a red circle. It's very efficient at reflecting red light, and so this is light compared to the rest. There's more light here than the rest. There's less light here than the rest. To make this equal that, we will turn down the amount of long wave light. What did we see? Well, this didn't turn black. Rather, everything got a little bit darker, but this is still light in relation to the rest of the field of view. Let's do it on the other side. This is an efficient reflector of green light. This is an inefficient reflector of green light. We must turn this down. And so we will do that. We start off with them being equal. We will turn them down. Again, what do we see? This doesn't turn black. This is still light in relation to everything else in the field of view. In all imaging and image reproduction used color separations. Printers in the 1700s used handmade color separations. Maxwell used silver halide separations. What Land proposed was that the separations are important. These were separations in light. What Land proposed was that it's separations in appearance. The neural interactions with areas interacting with each other. The neural interactions of the spatial information controls appearance. It's not the amount of light, it's the spatial comparisons. So we've used separations in 1700. Land's proposal was that this separation, when you change the amount of light, gave you this separation. Use this separation instead of the quanta catch of a single cell. Use 3 sets of spatial interactions to determine color appearances. So Retinex, the name of Land's theory, is a contraction of retina and cortex. He proposed that as much of the retina and cortex as needed to calculate 3 lightnesses, the L lightness, the M lightness, and the S lightness. And in fact, these lightnesses are the signal for color. Green is dark, light, dark. Red is light, dark, dark. Retinex has spatial comparison channels. So we make spatial comparisons to find the edges. We build all the edges up into an image in terms of lightness. We have three independent channels, L, M, and S. Each channel uses the edges to build the lightness. Color is simply L, M, and S lightnesses. So, the world of colorimetry, its cone quanta catch, it's the receptor information. It's all about the light-matter interaction. This is a necessary but not sufficient description of color vision. We follow cone quanta catch with long wave, middle wave, and short wave spatial records. We make spatial comparisons using the neural processing post-receptors to establish three lightnesses, and the three lightnesses determine the color. So the left side looked green, dark, light, dark. The right side looked red, light, dark, dark. Similarly, we can proceed through all the different colors, and color appearance is the result of three channels of independent lightnesses. We can test this by using an assimilation target. It's a very simple experiment. We have 9 identical red squares, here, here. We place them on different surrounds. They're still identical stimuli, but they don't appear to be identical. If we now look at this through a red filter, we see a long wave color separation. We note that this is lighter than this. That implies there's more red, more higher red lightness, implying more red in the appearance on this compared to this. We look at the green separation, namely look at the scene with the green filter. This is lighter than this. The right is lighter than the left. We look at the shortwave separation. Here, the right is darker than the left. Lighter implies more red. Lighter implies more green. Darker implies less blue. If we look up at the squares, the right-hand set of squares here appears more red, more green, less blue. The left side appears the opposite. It is less red, it is less green, and it is more blue. LMS lightnesses predict color appearances, and lightnesses predict color appearances in all situations. If we take the bottom-up quanta catch approach to color vision, Land has added the idea of comparing all pixels so we can continue along that path. We don't have to worry about adopting the more difficult path of discovering the illumination and discounting it. Appearance is not associated with the quanta catch, but it is associated with the spatial comparisons of quanta catch over the entire field of view. Now, there are plenty of other theories about color adaptation still active today. There is Von Trie's idea that the cells in the retina adapt, and it's the adapted quanta catch that explains color constancy. There's also many people who believe in the perception of objects. There are many people who believe in top-down processing. Which model predicts color constancy appearances in the high dynamic range seen? The answers to these questions is to ask the observers to make appearance matches. What do observers see with change in illumination? Does reflectance predict appearance? Does adaptation predict appearance? Or do the bottom-up long, middle, and short wave lightnesses predict appearances? We need to ask the observers. First, we'll test the Retinex idea that LMS lightnesses predict color. We'll use a simplified Mondrian. There are 3 parts to this. We will make a 17-area Mondrian shown on the right. These are the details. Measure the amount of light at the observer's eye, and also measure the appearances by asking observers to match things in the Munsell book. There's 5 parts to the experiment: gray, red, yellow, blue, and green. That goes for the 5 different areas selected for the observers to match and for us to measure the light falling on the object as well as the light coming to the eye. So we are going to vary the illumination and the reflectance to get a constant LMS radiance. So by changing the illumination and changing the paper, we get the same. Again, change the paper, change the illumination, we get the same. Change the paper, change the illumination, get the same, and so on. Observers match 5 areas that have constant L, M, and S radiances. In the next part of the experiment, we measure the appearances. The observer looks with one eye at the Mondrian with fixed uniform illumination, and the observer is asked to report on all 17 different areas. He chooses in a Munsell book viewed with the other eye. Each eye has independent adaptation, and this constant amount of L, M, and S illumination here. In all, there's 17 papers, 5 illuminants for 85 matches, and we'll portray the data using 3 different channels, both in the stimulus and the appearance. The observers report that despite the fact the radiances are identical, we get a very wide range of color appearances from the matches. The matches in the initial experiment, the gray experiment, gave us these Munsell chips. When we changed the illumination so each area had constant L, M, and S radiances, we got very similar, but slightly and systematically different choices of papers that match. The small, systematic departures from perfect color constancy. When we plot the Mondrian's radiance versus the matching paper's radiance, there is very little correlation between the cone quantic catch from these radiances to the cone quantic catch of the matching stimulus. So, first result, what do observers see with change in illumination? Mondrian papers have nearly constant appearances in varying illumination. Does reflectance predict appearance? Well, we have to step back and take a look at what we mean by reflectance. In a physics lab, it's a 3-step operation. We have some illumination falling on an object. That object is imaged onto a sensor, a carefully masked sensor, so it's only looking at one little spot. We get the radiance from the object in step 1. In step 2, we place a reference white in front of the object and make the same measurement. In step 3, we calculate that reflectance is equal to the radiance from the object divided by the radiance from the white stand-in. Vision can't do that. We don't have white papers that we walk around and place in front of each object to decide what it looks like. Instead, we replace the 3-step operation with edge ratios. We replace it by making spatial comparisons between the object and the white surround. Here is a spectrum. In physics, we measure the spectral reflectance one wavelength at a time. We scan through the spectrum and record one little piece of light at a time. That way we get the highest chromatic isolation. In vision, we use 3 receptors. We have 3 cone sensitivities. They have extremely broad spectral sensitivities. If we compare them, we see that not only are they broad, but they overlap. Here, if we can plot L and M on top of each other, we see that the major response is shared by both sensors. We have to be careful of the effect of cone crosstalk when we use spatial comparisons to calculate color signals. Remember this assimilation target that we talked about a few minutes ago. Here is the signal sent to the display in terms of that color. This is the red separation for this set of colors. You can see in the pure display, the signal sent to the red in the long wave record is the same as the signal sent to the yellow in the long wave record. So these are all the same, and these are all the same. However, when you include crosstalk in terms of the effect of the overlap of spatial comparisons, we can see when the perfect separation sees no difference between these, the crosstalk introduces changes in the samples of interest that affect the signal we are trying to detect. Here is a photograph of a still life scene taken with silver halide red color film technology. This is the spectral response of the red, green, and blue sensitivity curves. If we substitute human sensitivity curves, we get the considerable overlap we've just discussed. We see the crosstalk. In fact, in 1890, Ives of the Ives Metal and the Optical Society had a company that made color cameras. He filed a patent claiming that the best camera would have natural color and would use the spectral sensitivity of the eye. He used Maxwell's color matching functions and made filters that matched them and made and sold cameras. The problem was, because of the spectral overlap, the pictures weren't very good and the cameras didn't sell. Within 5 years, Ives discontinued this approach and went in the direction of isolated color spectral sensitivities. It's very interesting to look at these minimal between the sensitivities. It doesn't matter what the peak sensitivity is. What matters in getting good color results is where the minima are between the sensitivity curves. Measure the light at the eye's cornea. We can measure the light that comes from the scene, but we're really more interested in measuring the LMS radiances from all 17 areas as the receptors see them. In order to do that, we need to calculate an integrated radiance. Namely, we need to calculate the radiance from the light, these 3 narrowband emitters, with the cone sensitivity curves to get 3 integral values, the long, middle, and short wave integrated radiance. Next, we really want to work in a uniform color space. Munsell did a very interesting bisection experiment. He had a black and a white. We repeated the experiment using 3 log units of dynamic range. Munsell's experiment was to ask the observer to pick a chip that was halfway between white and black. Then he bisected it again, 5 and 9 to get 7, 5 and 1 to get 3, and so on, to make 9 equally spaced steps in appearance. If you take those chips that the observer selected and plot them against log luminance, you get a cube root function. The cone response is log retinal luminance. So we can convert actual luminance to receptive response by using a cube root function. There are many details to this, which I won't share with you at the moment, but I have provided a detailed set of references so that if you're interested in this, you can follow it at much greater depth. To make a long story short, we are going to introduce the idea of a scaled integrated radiance. That is, we take the light, we take the cone sensitivity, and we scale it by a cube root in order to get a number that represents L, M, and S cone responses. We can use these scaled integrated radiance values to calculate the scaled integrated reflectance. We take the scaled integrated radiance that comes from the object, divide it by the scaled integrated radiance that comes from the surround, and we do it 3 times. When we compare the integrated reflectance of the Mondrian chips in each waveband against the scaled integrated reflectance of the matching area in the Munsell book, we find excellent correlation. Mondrian reflectances using scaled integrated radiances correlates with observer matches. Final question is, can you use this in a model to predict color? Complex scenes have many edges. What we described was a way of calculating a scaled integrated reflectance of an object in a uniform surround. But in a real scene, we don't have a uniform surround. We have to build it up from the edges. Here's a simple display. It has 16 different papers, but if you count the edges, you see that there are 32. The Retinex model calculates all scene elements using all pixels. Again, that is a big topic, so I'll just leave a reference. The result in the McCann, McKee, and Taylor experiment is that the observed likenesses can be calculated by Retinex computed likenesses. We have L, M, and S results. Here we have observed likeness. Here we have computed likeness. And this is a magnification. You can see that the correlation is very good. The results of this experiment says that the data supports spatial comparisons. Retinex models predicts observer matches. There are small systematic departures from perfect constancy. Cone color crosstalk causes the departures from perfect constancy in these experiments. Following the bottom-up approach to color vision, Land introduced the idea of comparing all the pixels to calculate likenesses. The McCann, McKee, and Taylor experiments provided quantitative verification. Bottom-up spatial comparisons do not require object recognition in order to explain color constancy. Does adaptation predict appearance? Moncrief proposed an adaptation theory to get back towards a bottom-up calculation of color appearance. Now, all neurons adapt. The neurons in your retina adapt, and the neurons in your feet adapt. Vision adapts to light. Dramatic changes in threshold sensitivity, more than 10 log units. Dark adaptation is slow. It involves the regeneration of photopigment. It takes place in the receptor and in the pigment epithelium cells. Light adaptation is fast. It is neural. Does adaptation affect color constancy? Does adaptation make a difference in a complex scene? We know that stabilized images where by some means you hold the image perfectly steady on the retina, they just disappear. Eye movements are necessary in order to see real scenes. After images are shut off like complex scenes. The edges must fit. Two of my friends, Nigel Dorr and Gabriel Marcoux, have done some extraordinary work in this area. Nigel published a paper in 1962, Why After Images Are Not Seen in Normal Circumstances. And that's why Land's Mondrians have irregular shapes. If Land used a checkerboard, he realized from Nigel's experiment that the after image could carry from one square to another. By using rectangles that didn't superimpose, Land removed the effect of after images in his Mondrian experiment. Gabriel Marcoux, who's worked for Apple for many years in display calibration. Gabriel has done remarkable work in calibrating Apple monitors in the lab and in manufacturing. He has some experiments I want to share with you about color and after images. At the tip of the arrow is a fixation point. I'm going to ask you to stare at that fixation point and to continue to stare at it for several minutes. I'm asking you to form an after image of the color scene. Stare at it, don't let your eyes move. Keep it fixated right at the tip of the arrow. In a moment, I'm going to change the scene. I'm going to ask you to keep fixated on the point. Keep fixated and count to 3. Then look off to the corner of the image. So just to repeat, when I change the image, count to 3, stare at the same point. Then I'm going to change the scene. I'm going to ask you to continue to look at the fixation point till I count 3. Then I'm going to ask you to move your gaze to the corner of the picture. I'm going to change the picture now, so continue to look at the fixation point. I'm changing the picture. Continue to look at the fixation point. Continue to look at the fixation point. Now look around the scene. We have a scene. We have a fixation point. That forms an image on our retina. The receptor cells continue to send the signal out of the retina until they are dark adapted. When we look at it, we don't see the positive after image. As long as you're looking at the fixation point, you do not see it. We then change the scene to a black and white picture of Gabriel. The after image is still there, and as long as you're fixated in the same spot, you see a colored version of the scene. When you look around, the after image transferred with the fixation point. We didn't see a colored image, we saw a black and white image wherever we looked, anywhere in the field of view, except, of course, the original fixation point. After image is fixed in the retina. It follows the fixation point. When the edges of the after image don't fit the edges of the incoming live scene that we're looking at, it distracts from the image, it does not add to the image. Vision turns off the after image when the contours don't fit. We're going to repeat the experiment with a new scene. Again, please stare at the fixation point at the center of the image. Stare at the image. The better you fixate, the better your results will be. Don't rotate your head. You can blink. The better you focus on that single small point, the better your after image will be. It will be sharper if you can maintain constant fixation. So, we're going to stare at this for about a minute. Continue to stare at the point. Continue to stare at the point. Don't rotate your head. Your eye is taking a picture. Don't look at your cell phone. Stare at the point in the picture. When you stare, you are adapting the rods and cones in the retina. The eye adapts and makes a temporary record in the rods and cones. What happens to that temporary record? Does adaptation control color constancy? Or do we have a mechanism that shuts off adaptation when we look at a complex image? Continue to look at the spot. As before, I will change the picture. I will tell you when I'm going to change the picture. I present the new picture. I want you to look at the point and count to 3. After that, look around the picture. Look all around the picture. Look back at the spot. Look around the picture again. Look back at the spot. Look around the picture again and back at the spot. The color in the image fades as the eye dark adapts and removes the temporary record in the rods and cones. We see color after images when the edges fit. Adaptation makes after images. As I said, all neurons adapt and vision adapts to light. Does adaptation affect color constancy? Changing uniform illumination, as we did in the previous MMT, must change color adaptation levels. Does adaptation level affect color constancy? Or does color constancy act as after images? Here's the challenge. Can we cancel adaptation? Can we shut off color constancy in a complex scene. We know that light from the scene causes adaptation. When we change LMS illumination, we change adaptation. Can we remove adaptation by changing the scene? We're going to repeat the previous MMT experiment with a large gray surround. We're going to measure the radiances from a gray paper, LMS, and then we're going to measure the average radiances from the entire Mondrian. In step 2, we're going to adjust the illumination so that a red paper, instead of a gray paper, sends LMS to our eye. We're going to measure the average from the entire field of view. In the third part, in the experimental part, we want to replace the surround with a new surround that changes the average. We measured 1,000 papers and selected the best 5 in order to repeat the McCann, McKee, and Taylor experiment with no adaptation. In step 3, we use the illumination from step 2 and change the surround so that that combination that moves us back to having AVL, AVM, AVS, the same average as in both step 1 and step 3. So with constant average values, we're going to do the gray, red, green, yellow, and blue experiment. We're going to have constant average values, and we're going to have constant stimulus values for the area in question. We're going to do this not only in the big uniform surround, but we're going to take the Mondrian apart so we have a maximum coverage of the local surround. The experiment removes adaptation stimulus, and so an adaptation theory predicts that all L, M, and S samples, each of these 5 areas that look different colors in the McCann, McKee, and Taylor, now must all look the identical gray. To recap, we're going to compare the results from step 1, which is McCann, McKee, and Taylor, and step 3. There are equal L, M, and S values coming from the gray paper here and the red paper here. There are equal average radiances from both in both experiments because the reflectance canceled the effect of the illumination. What does it look like? Changing the average back to the original average should make the gray paper look gray and the red paper look gray as well. Step 3 is supposed to cancel adaptation. Despite identical cone quanta catches and scene averages, paper have different sensations, gray and red. Constancy ignores adaptation averages. Here are the results from all the experiments. We have the 5 different colors. We have the actual color specification in the Munsell book. We have the matching chip from the MMT. We have the matching chip from the surround experiments. We have the matching chip from the local surround experiments. And we have the calculated MMT values. As you go across any of these colors, you'll see that there's very, very little change in color appearance. Even though we have worked very hard to cancel the adaptation stimulus, we find that colors are essentially constant in these Mondrians. Chromatic stimuli have minimal effect on appearance. Retinex model calculates these appearances. We do not find support for the idea that von Kree's adaptation is the explanation for color in these Mondrians. But wait, there are more adaptation theories. In 1988, Nayatani proposed incomplete adaptation. This is the mechanism that's central in the CCAN model of color appearance. What limits the color constancy? Cone crosstalk or incomplete adaptation? We performed an experiment where we matched 3 papers in 27 different illuminations. These are the description of the papers. The illuminant consisted of 3 sets of 4 LEDs. There were a blue, a green, and a red LED, and we could turn on either 4 or 2 or 1. If we turned on 4 reds, 1 green, 1 blue, it fell here in CIEXY plot. If we turned on 4 greens and 1 blue and 1 red, and so on. We asked the observers to match those 3 papers and the 27, that is the all possible combinations of 4, 2, and 1 LEDs. If we plot this in an LED 3D space, we see that these illuminants formed a cloud. There's 27 points in the cloud. We're going to consider 2 hypotheses, chromatic adaptation and spatial comparisons. The first studies the idea of discounting the illumination. The second works on objects, namely the spatial comparison of objects. It responds to the object's ratios of quanta catches, it's bottom up, and the spatial comparisons control the predictions. It requires the entire field of view. Chromatic adaptation models, however, respond to the illumination, but the illumination values are given as an input, and these models consider only the single color sample of interest. Nayatani used an analysis of the MMT data to quantify incomplete adaptations. The idea is if we have a yellow paper and we change the illumination to make it equal to a gray paper, the eye adapts and comes back along the change in illumination to a value close to the original paper. Nayatani uses an analysis of MMT to quantify incomplete adaptation in CID. The idea is that it responds to the illumination. So in MMT, when we had a yellow paper, we changed the illumination to make the light coming from it be equal to a gray paper. Nayatani's incomplete adaptation says that it doesn't come all the way back to the original, but comes back most of the way. In fact, the model that Nayatani calculates is here. The data from MMT is here. So the spot in the center is the result. The tips of each line is the starting point of our 27 different illuminations. The solid ball is the result of incomplete adaptation predictions. Experiments use a constant array of reflectances. Changes in red, green, blue illumination spectra causes changes in the LMS sensitivities. This is a global shift. It's the same for all papers. That means the 27 illumination predictions for these three papers makes 3 clouds of appearance predictions. Spatial comparisons use ratios. It builds up L, M, and S likenesses. We've already considered the overlap of the spectral sensitivity curves in the eye. So the 27 illuminant, the numerators and denominators are changed by the same factor in the illumination, but the ratio of radiances is nearly constant. It has imperfect constancy because of the integrated reflectance changes from cone crosstalk. It synthesizes nearly constant appearances. Crosstalk is 0 with grays. It is significant with high chroma colors and intermediate with low chroma colors. It depends upon the paper's reflectance. This is the predicted appearances using ratios with crosstalk. We don't get a cloud. We see that the predicted changes in appearance from crosstalk are affected by the overlap of the cone spectral sensitivities, So, we have here the predicted 3 clouds from a model discounting the illumination. And we have 2 lines and a point. These movies, particularly running 3 of them simultaneously, challenge the bandwidth of the video recording. I'm going to switch to still life representations of this data. You've seen in the movies the cloud-like nature of the incomplete adaptation illumination-based predictions. At the center of this cloud is the predicted appearance in achromatic illumination. Around it are all the 27 different colors predicted by incomplete adaptation. At the other side, using the pale purple paper, we see a second cloud and a third cloud around the L axis at the center of the LAB space. These are the predictions made by incomplete adaptation. Using crosstalk in the calculated appearance produces very different results. First, let's look at the gray. Crosstalk is 0 for achromatic reflectances. Highly saturated colors predict a long line in LAB space. The less saturated purple paper is a shorter line in LAB space. When we ask the observer to match these, we find this excellent correlation between the observer data and the predicted appearances based on crosstalk. Departures from perfect constancy do not correlate with discounting illumination. Crosstalk predicts color matches when we use 27 different illuminations. The limits of color constancy are physics-based. It's all about stimulus and sensitivity of the cones. It's not illumination dependent. It's not incomplete adaptation. It's not framework controlled. It's not perceptual. It's not cognitive. It's not top down. It's just physics. So In this section, we have verified the bottom-up spatial comparison model can work for color Mondrians, and we have found no evidence to support von Kree's adaptation, incomplete adaptation, or top-down object recognition. We have 3 more topics to finish in this talk. This is a good time to pause if you would like to take a stretch, but do come back. There's 3 interesting things to discuss. We all know about gray world and about the average for a photographic scene. But in fact, vision is more interesting. It normalizes the scene to the maximum. You'll see this in Maximoff's shoebox experiment. Does color constancy correlate with the surface reflectance in real scenes? That's another very interesting experiment and a final summary. So join us back at the slides very soon. Around 1990, I met the distinguished Russian scientist Vadim Maximov. He studied the color vision in animals. He did some remarkable work. He shared with me an experiment that he and a student were working on, namely his shoeboxes. The idea was very simple. He made an array of papers and placed them in a shoebox to be viewed through a hole. He had a filter on the top of the shoebox to provide a spectral illuminant on the diffusing box to illuminate the papers at the end of it. Maximov's experiment was to find if edges controlled color appearance. He believed that if he could find a set of matches and a filter that produced a particular set of radiances, 5 different radiances in this case, that were identical to a different set of reflectances in a different illuminant. Maximov was, in 1992, was limited in terms of the availability of digital printers. We happened to have a Canon copy machine that had a very primitive digital interface, and we were able to make papers that had any amount, yellow, magenta, cyan, and black pigments. We could control digitally the amount of pigments deposited on the paper. Only with such remarkable control can you get papers that have the desired property. Maximov's idea was that when we shifted from one light to another, it would change the illumination on all of the papers to the same extent. We made papers that had constant edge ratios that shifted in color space. So the papers shifted color space in one direction, and the illumination brought them back to the same point. By controlling the edges and the illumination, we arrived at a set of 5 different positions in color space, 5 different quanta catch. With these papers and this illumination, we were able to get to the same 5 quanta catches. What did we see? The stimuli were identical, but as we all know, color constancy plays a big role when we look at a complex scene. Maximov's prediction was correct. Constant quanta catch everywhere in the field of view generated equal appearances. Constant ratios in different illumination produce the same set of appearances. The second experiment was even more fun. We added 2 identical white bands. Everything else was the same. So the quanta catch for all 5 areas was identical, but we changed only the surround. It restored color constancy. There was a slight shift. You can see this is a little more cyan and this is a little more red. But equal quanta catch did not appear the same as long as they had a common maxima in the field of view. So, if you adjust the illumination and the reflectance in a complex scene, you can get appearances to be the same and shut off color constancy. When you introduce a new maxima, it can be a white, or it can be a new maxima in any of the 3 channels, the L or the M or the S maxima, will restore color constancy. So the maxima plays a very important role in the appearance of complex scenes. The second set of experiments is measuring color constancy in high dynamic range scenes. We call this a 3D Mondrian. It's made of a set of blocks. All blocks, in this case, are painted with exactly the same paint. The difference is the illumination. When we use directional illumination, we have sharp edges in the illumination. Again, we made 2 identical sets of color 3D Mondrians. Each block was painted with one of 11 different paints. So the surface reflectance of all the reds in both scenes on the blocks is identical. There are about 200 facets in these blocks. These are the paints used on all the facets in the blocks. Karina Perriman from Bristol measured the low dynamic range and the high dynamic range appearances of these blocks. She did it by making 2 paintings with watercolors. These watercolor paintings of the appearance of both LDR and HDR scenes, the watercolor appearances matched the scene's appearances. If we measure the watercolor's reflectance for each facet, color reflectance quantifies the appearance of the facet. So we have 2 illuminations, the LDR and the HDR. We're going to look just at the red blocks. These arrows indicate all of the sides of the blocks painted with red paint. When the illumination is almost uniform, the reflectance curves measured from the paintings that reproduce the scene are almost the same. When we use directional illumination, Karina painted all these red surfaces with different watercolor paints. If we look at all 6 of the paints used on the blocks, we can see the same pattern. In nearly uniform light, for red, green, blue, cyan, magenta, and yellow, there is considerable overlap. When we increase the number of edges by using directional illumination, we can see the color constancy in the HDR is much poorer. The result is appearance does not correlate with reflectance. Rather, appearance is the spatial comparison of quanta catch. The question was, do color appearances correlate with surface reflectance in real scenes? That was Helmholtz's hypothesis. Some people attribute it to Land, but Land didn't believe it at all. We chose these blocks because there's no reason to believe that any of these surfaces should have a particular color. The answer to the question about reflectance and appearance depends upon the scene. When we went to the 3D Mondrian in nearly uniform light, most of the reflectances correlated with the appearances. When we used high dynamic range light on the surfaces of these blocks, we found very poor correlation between reflectance and appearance. We can think of this as the correlation with edges in illumination. When we have flat and uniform display where there are no edges in illumination, we get excellent correlation. That correlation erodes when we go to 3D objects in low dynamic range illumination. However, when we go to high dynamic range, where there are many edges in illumination, we find the correlation between reflectance and appearance is very poor. Constancy is poor in LDR illumination and bad in HDR illumination. We see this in the real world as well. Edges in illumination change the appearance of objects. This is a set of pictures taken over a period of about an hour in Bryce Canyon, Utah. Bryce is at 9,000 or 3,000 meters elevation, and there was a winter storm coming. So the clouds were moving rapidly, and the shadows changed dramatically as you watched. The appearance of these rocks was far from constant over that hour. Another example of a cloud creating a difference in appearance, the shadow of a cloud creating a change in appearance of uniform reflectance objects. This is my favorite. These are two pictures taken 75 seconds apart. This time a cloud got in the way of the sun. There is a very large change in appearance of objects with perfectly constant reflectance. However, this scene had substantial edges in illumination. I'd like to summarize now. Edges in illumination have the same effect on appearance as edges in reflectance. As we saw in the previous TPU symposium talk, edges have maximal influence on appearance. A scene segment with LMS quanta catch can appear any color. Chromatic adaptation stimuli have minimal effect on appearances. Departures from perfect constancy correlate with crosstalk, not with incomplete adaptation. Rods and cones shows chromatic adaptation. We see adaptation in after images. But without matching edges, vision shuts off the adaptation signal in complex scenes. I've done research on dock adaptation since 1963. Adaptation is a big effect. It has a 1,000,000 to 1 change in light sensitivity. I've been measuring the amount of chromatic adaptation in complex scenes since then. This talk described many of these studies. I never found a significant effect of chromatic adaptation in scenes. The reason is that vision has a mechanism that blocks the adapted cone outputs sent out of the retina, unless the edges of the after image fit the edges of the instantaneous scene on the retina. I never found chromatic adaptation in scenes because spatial vision shut off the adapted cone signals. Scene maxima control color appearance. We see this when we go to the trouble of shutting off color constancy. We used 2 complex scenes with different reflectances. We used illumination differences that cancel that. With exactly the same quanta catch from these 5 areas, the appearance of the 5 areas is the same. We have effectively shut off color constancy. However, when we add a white stripe or a new maxima of any of the LM&S channels, we restore constancy. It's not perfect constancy, there are small differences, but a new maxima restores constancy in a complex image. Object reflectance and appearance. The correlation is scene dependent. In flat, uniform displays in uniform illumination, we always find a high correlation with object reflectance. However, in this scene, all the information is represented as edges in reflectance. In 3D Mondrians, color constancy erodes. When we go to high dynamic range illumination, we see a serious erosion of color constancy. We measured the observed lightness in the L, M, and S channels. We found that we were able to calculate the appearance that matched the observed appearance. We did not find support for the following hypotheses. Cone quanta catch alone does not predict color appearance. Cone quanta catch is essential, however, in measuring edges. Adaptation does not discount illumination. Adaptation is real, and as we saw with after image, is significant, but it does not discount illumination. Observers are very good at recognizing illumination. However, that skill does not explain color constancy. Observers do not see an object's surface reflectance. They are very good at recognizing it, but that recognition is not the key to color constancy. Observer data supports only one theory. Edges in reflectance are the same as edges in illumination. Independent LMS channels calculate LMS lightnesses. cone quanta catch generate receptor responses. It's the neurons that make spatial comparisons that generate the LMS lightnesses that control color. Edges cause large changes in lightness. Gradients produce very small changes in lightness. There are additional topics not included in this talk, but are the subject of other talks, namely, measuring the light at the cornea does not tell you the light on the retina. Intraocular scatter plays a major role in vision. The action spectra of LMS quanta catch is not equal to LMS lightness. The overlap of the cone spectral sensitivity responses show the need for chroma enhancement, similar to that used in electronic imaging. Chroma enhancement improves color separation and minimizes crosstalk. Landon McCann Retinex models have been used in photography. They convert camera digits to lightness. They improve HDR scene rendition. When visiting Karina Paraman one day, she took me to Fox Talbot's estate, where he made the first surviving negative photograph. He did that in 1835. That photograph was a photograph out this window. Karina and I made two more pictures. One, we used the tone scale mapping that has dominated color photography from 1945 to the present. That's the central picture. The picture on the right was made in a camera using Retinex processing, a process that builds images up from edges and integrates those edges over the entire field of view. So what do you think? Are you a fan of chromatic adaptation, or are you a fan of spatial comparisons? I'd love to discuss this with you and look forward to it. I'm a great admirer of the beautiful science that has led us to the quanta catch of the cone pigments in the receptors in the retina. While quanta catch is essential, it is the ratio of quanta catches that is necessary to predict color in the natural scene. Behind this link, you will find copies of this talk, the earlier talk of the TPU symposium, and many links to the papers that support this point of view. I look forward to talking with you. Contact me at mccans@tx.net. And thank you very much for your patience. And I hope to talk to you soon. Bye now.